right, so that's pretty much what you need to know about replication. We'll talk a little bit more in the last segment about what happens at the very, very end, but you've got the basic idea. You begin at a origin, the DNA separates to make this bubble, and there's replication forks on each end of the bubble where DNA polymerase, along with its helper molecules, builds DNA with lagging strand synthesis on one strand and leading strand synthesis on the other. So let me get to some of the historical guys who have helped us understand DNA and how replication works. So the first people I wanted you to know are three guys named Avery, McLeod, and McCarty. And their work was the reason that Watson and Crick cared enough, they were interested enough to figure out the double helix shape of DNA. Avery, McLeod, and McCarty are the guys who first demonstrated that genes are made of DNA. And they did this with bacteria, which not too many people cared about bacteria at this time in genetics, but it turned out the bacteria are really great to study genetics with. So Avery, McLeod, and McCarty had this situation where we had bacteria that we knew were picking up a new gene and it was changing them. So that process where bacteria get a new gene and they can do something different that they couldn't do before, that's called bacterial transformation. And Avery, McLeod, and McCarty took the transformation process and figured out, they took that liquid that they knew that liquid somehow contained genes in it. And they took that liquid and one at a time, they found enzymes that would chew up everything that was in that liquid, see if transformation still happened. They found that if they added an enzyme that digested protein, that didn't stop transformation from happening. So that meant genes weren't made of protein. They had an enzyme that would chew up RNA, and they added that. And they found that even after they chewed up all the RNA in the sample, you could still get transformation. So the genes weren't made of RNA either. And then they also had an enzyme that would hydrolyze or chew up DNA. And when they added that enzyme, the DNA enzyme, to their sample, that prevented transformation from happening. So that was evidence, the first experimental evidence that genes are made of DNA. And they won a Nobel Prize for that. Prior to that time, actually, I think most people thought genes were made of protein. So it was kind of a surprise that DNA was a genetic material. Another fellow who contributed to our early understanding of DNA and who actually kind of discovered the base pairing rules, although he didn't recognize what he was discovering, was Erwin Chargaff, who came up with Chargaff's rules. So he studied DNA from a variety of sources. And what he found with the DNA is he found that DNA from different organisms had different proportions of A's, G's, C's, and T's. He measured very precisely the proportions of those and saw that there was a pattern, regardless of the specifics of the percentage of A's in DNA samples. He found these patterns. He found that the proportion of purines was always equal to the proportion of pyrimidines. That is, if half of the bases were, half of the bases would be A's and G's, and then the other half of the bases in the DNA would be C's and T's. That was true whether it was 20% A or 40% A. Either way, no matter what, the A's and G's added up to 50%, the C's and T's added up to 50%. And that makes sense to us now because we know every base pair has one purine paired to one pyrimidine. The other thing he figured out was that whatever the percentage was of A's, the percentage of T's would be equal to that. And whatever the percentage of G's, the percentage of C's would be equal to that. And that now makes sense to us as well because we know when there's an A on one strand, there's a T across from it. Where there's a G on one strand, there's a C across from it. So Chargaff wasn't at the point where he understood the double helix structure or the fact that you had A's and T's base pairing with hydrogen bonds and G's and C's hydrogen bonding together. But he did figure out this pattern, and it helped Watson and Crick a lot when they were figuring out their double helix structure. So it was an early clue to complementarity or the base pairing rules. This is Hershey and Chase. Marcia Hershey and Chase, they kind of, their results supported Avery McLeod McCarty's, but they were working with different organisms. So Hershey and Chase did their work at a very famous molecular biology lab in New York State called Cold Spring Harbor Labs. And the experiments they did, like many people at Cold Spring Harbor at the time, they were doing experiments studying a virus that could grow in bacterial cells, E. coli bacteria in particular. So these viruses that grow in bacterial cells are called phage or bacteriophage, and they're safe to grow. They're pretty safe to grow in the laboratory. They're pretty easy to work with. And Hershey and Chase were able to do these experiments because there had just recently been World War II, where nuclear weapons were developed. And one effect of that was a lot of the development was done at Oak Ridge Laboratories in Tennessee. And as a sort of a waste product of their weapons research, they were producing a lot of radioactive waste, including radioactive isotopes of sulfur and radioactive isotopes of phosphorus, which turned out to be super useful for studying molecular biology and molecular genetics. So Hershey and Chase actually got the material to do their experiment from Oak Ridge Weapons Lab. It's near Knoxville, Tennessee. So in their experiment, they had these viruses, and these viruses are very simple structures. They have DNA and they have protein. And we knew about viral reproduction that during a virus infection, the genes of the virus have to get inside the cell. So Hershey and Chase investigated what exactly was getting inside the cell. We knew genes were getting in, but was it the DNA or was it the protein? And they were able to use the radioactive sulfur and the radioactive phosphorus to mark each of those molecules so they could keep track of it. And the radioactive sulfur, remember that proteins will have sulfur atoms in them sometimes, so they could grow virus in cells that were growing in food with radioactive sulfur. And then the baby virus that you got out, their proteins would be radioactive. You could take another batch of the virus and grow them in E. coli that you were feeding with radioactive phosphorus isotopes. And if you did that, then the baby virus you got out of that culture would have radioactive DNA. So the phosphorus would mark the DNA, make it radioactive so you could track it. And the radioactive sulfur, the S35, would make proteins radioactive so you could track them. So they had two preps of virus, one where the proteins were radioactive and the other where the DNA was radioactive. And they did this thing that's called the Waring blender experiment, or just the blender experiments. They take the virus and then mix it with some E. coli cells long enough to give the virus a chance to infect the cells, to let the genes go in. And then, so when that happens, the virus attaches to the outside of the cell, and then the genes go in. After they had allowed the virus to start the infections, they used a blender. They stuck what was in their tube into a blender and whirled it around, and the blender knocked off the rest of the virus. So the genes were inside the cell and everything else got knocked off by the blender. And what they discovered when you did that is that if you started with virus where the DNA was radioactive, the DNA stayed in the cells. After you blenderized, the DNA was still in the cells. And if you used radioactive protein, the S35 labeled virus, then after you blenderized, the cells weren't radioactive. They had the genes in them, but they weren't radioactive. And the liquid, the rest, when you pulled the cells out, the liquid had the proteins left behind. So that was more persuasive evidence that genes are made of DNA, but this time it demonstrated that viral genes are made of DNA. So Avery, McLeod, and McCarty showed bacterial genes were made of DNA. Hershey and Chase showed that viral genes were made of DNA. We're starting to see a pattern here where organisms' genes, regardless of the organism, seem to be made of DNA. So that was more of the information that had Watson and Crick so interested in figuring out what DNA looked like, since it's what organisms seem to be making their genes out of. Coming up with the double helix shape, like I said, it was done by taking a lot of information from a lot of different experiments into account and building a model of the molecule that would fit all of everybody's observations from all of their experiments. A couple of people who really contributed very, very strongly to Watson and Crick's breakthrough were Franklin and Williams. And so Rosalind Franklin and Maurice Wilkins, she worked in his lab, had studied DNA, purified DNA, by shooting X-rays at it and measuring the angles that the X-rays would bounce off the molecules. And they saw this kind of an X-shaped diffraction pattern that suggested it was a helix. And then Watson and Crick were able to take that along with other pieces of information, including, there was another woman who was a sort of a biophysics person like Franklin and Williams, who actually gave them, she also gave them quite a bit of a push towards the A, T, G, C base pairs. But at any rate, they took that experimental data from lots and lots of different people doing different sorts of experiments and built that double helix model that took into account all of those observations. Okay, I already told you that DNA replication is semi-conservative. I said you'd need to know the guys who demonstrated that, and it was Meselson and Stahl. Meselson and Stahl also worked at Cold Spring Harbor Labs. They were working with E. coli, not with viruses. But they did a cute experiment that showed that DNA replication was semi-conservative. And they used isotopes like Hershey and Chase did, but they used a different sort of isotope. They used isotopes that were not radioactive, but isotopes are always different in mass. So they used two different density forms of nitrogen to do a study of DNA replication. Now, the A's, G's, C's, and T's in DNA contain a lot of nitrogen. They're called nitrogenous bases. So what kind of nitrogen is in the DNA has a big effect on the density of the DNA in a polynucleotide strand. So they took some E. coli bacteria and they grew them in food where all of the nitrogen was a heavy isotope of nitrogen, nitrogen 15. They grew the E. coli for generation after generation after generation until pretty much all of their DNA had just that heavy nitrogen isotope. If you took those E. coli cells that had been growing for a long time in heavy nitrogen food, cracked the cells open and got their DNA out, you can do density gradient centrifugation with DNA. And you can see how dense the DNA is by how far down the tube it goes during density gradient centrifugation. And so after growing in heavy nitrogen containing food for a long time, the DNA that Meselson and Stahl got out of their E. coli was really dense, so it went really far down the tube because the densest part of the liquid in the density gradient tube is going to be down here. So the heavy DNA goes to a region that matches its density, which here is very low. They switched over, they took some cells that they had grown in the heavy nitrogen for a long time so that their DNA would all look like this picture on the left. And then they took those cells and they took them out of the food and washed them off, and they put them in fresh food that contained only the light isotope of nitrogen. And they gave them long enough for those cells to all copy their DNA one time. So they replicated one time. And then they cracked those cells open and got their DNA out. And they found that all the DNA that they got out of those cells was lighter than the DNA of the cells that had just been grown in heavy nitrogen. So you didn't have a really, really dense band at the bottom of the tube anymore. After just one replication, all of the DNA was now migrating to a lighter position in the tube. And then they took some of those cells that had grown for one replication of their DNA in the light nitrogen, and they let them go again. They let them do 2 replications of their DNA. And when they did that, they cracked open that group of cells, put their DNA in a density gradient, centrifugation. Then what they found is they still had some of that sort of medium density DNA that they had had after the first replication, but now they had a new band that was close to the top of the tube. So what this indicated is originally the E. coli, after growing a long time in heavy nitrogen, had 2 strands of DNA that were both heavy strands. That's the original DNA, had the heavy strands. And then after a replication round, each of those heavy strands got used as a pattern to build a new strand with the light nitrogen. And so those double helixes you got after one replication, each of them had one of the original heavy strands and a brand new light strand, shown as the dark blue line in this double helix. So that's going to be 1/2 is heavy, 1/2 is light. So it's kind of a medium density overall. When this DNA went through another round of replication, when this DNA pulled apart, one of the strands is still all heavy, and it's going to be a pattern for building a new strand that's going to be all light, because all the food that the bacteria are growing in are light. So that's going to still be at that medium density in the gradient. But then when this double helix pulled apart, the other strand in this double helix started out all light. And then when it was replicated, the new strand that was made using this dark blue strand as a pattern was also all light. So that's why half of the DNA after your second replication, half the DNA was medium density and the other half of the DNA was the light density. If you allow these cells to go through additional rounds of replication, you get a higher and higher proportion of your DNA migrating up here at the top, the light density, but you'd still maintain that intermediate density that was maintaining that original heavy strand. So it's semi-conservative because you're keeping that old strand, the heavy strand here, even while you're producing new light DNA. Okay, that's the end of this segment. And then the last segment will be mostly just kind of checking up and reviewing on that content. And then we're going to add a little bit more about telomeres and telomerase and what happens when replication gets to the end of a eukaryotic chromosome.